Good morning, afternoon, evening, or good night, depending on when you're watching this video. This is Mr. Messina broadcasting, and we're going to talk about synthesis organelles. In the previous video, we discussed the structural organelles, which actually help a cell maintain its shape, keep things out, keep things in, but still allow things to come and go. This time we're going to be talking about the synthesis organelles, and the synthesis organelles are the individual organelles that actually make things. There are four different types of synthesis organelle. There's the nucleus, which is the first thing we're going to talk about, ribosomes, the endoplasmic reticulum, and the Golgi apparatus. Now, the first and arguably the most important one is the nucleus. The nucleus, it's a compartment within a cell that stores the DNA. DNA is short for deoxyribonucleic acid, which is a very long phrase, but it's one of the most essential things in any form of eukaryotic cell, primarily because the DNA is what controls protein expression and thus controls what exactly the cell can and cannot do, what structures it has, how it looks, things like that. So this nucleus holds all this DNA inside of it, and it has a barrier that actually prevents the DNA from escaping, and this is called the nuclear envelope. All over this nuclear envelope, we have little pores called nuclear pores that actually allow some of the DNA to escape in the form of RNA, which is a little bit different, and we'll talk about it when we get to protein synthesis. Now, a nucleus looks actually like this. See, you got your main cell all around here, and then right here in the dead center, there's the nucleus. Now, in actual cells, the nucleus isn't always dead center, but it's sort of in the middle. Over here, if you actually look at this kind of blown up picture of the nucleus itself, you can see the nuclear envelope, which like the cell membrane is this bilayer that keeps some things out, some things in, but it still has nuclear pores, which allows some things to pass in and out of the nucleus. Now, that's not a very long list. Only a few things can make it into the nucleus, and only a few things can make it out of the nucleus. Now, within the nucleus, there's like a smaller nucleus called the nucleolus. This is actually the site where ribosomes are made, and we're going to talk about ribosomes in just a second. I also want to bring up the fact that this right here and that right there, those two are actually not part of the nucleus. This whole thing right here is the nucleus. This is the Golgi apparatus, which we'll talk about, and the endoplasmic reticulum, which you guessed it, we'll talk about. Now, this is a nice drawing, a nice cartoon that's clear and clean. In an actual cell, the nucleus looks closer to this. You can see right here is the nuclear envelope, which is like a cell membrane that keeps things out and keeps things in. Right here you can see the pores, right where the dye has stained it kind of like an orange color. You can see the pores. And the nucleolus is right here. It's this dark kind of purple, somewhat twisty looking thing in the center. This is where ribosomes are made, like I said before. Now, the nucleus holds all the DNA, but the workhorse of the cell that actually produces the proteins are the ribosomes. They're cellular structures that, like I said, they produce proteins. They look crazy interesting, primarily because they're made of two different pieces. You have a top large piece, which is right up here, and then you also have a small subunit down here. So whenever a protein is going to be made, you see a piece of RNA kind of slide between these two pieces, these two big pieces of the ribosome, or the big piece of the ribosome and the little piece. And then the protein is actually built, the peptide chain is actually built here. We'll get more into it when we get to protein synthesis, but just remember, ribosomes are the cellular structures in which proteins are made. And it's kind of important because if you don't have a firm grasp of what a ribosome does, a lot of other things may not exactly make sense. Now, ribosomes, like I said before, are kind of free-floating. They can actually hang out in the cytoplasm. The endoplasmic reticulum is actually kind of a wall that has ribosomes embedded into them. So ribosomes can either be free-floating or they can be attached to the endoplasmic reticulum. So it's a huge system. It's almost like a maze or a labyrinth, where RNA can go into this labyrinth and come out the other side as a full-blown protein. So there's not only just proteins that are manufactured, you have two types of ER. So you have the rough ER, or the rough endoplasmic reticulum, which helps transport proteins, that are made by ribosomes. Pretty much what we were talking about before, but you also have the smooth ER, or the smooth endoplasmic reticulum. And they don't actually have ribosomes attached to them, and they produce lipids, which are oils, as well as steroids. And steroids are special molecules that I'm sure you've heard about in the news, where some athlete or somebody has been using steroids and now they're really big and muscular. That's sort of kind of right. Actually, steroids are they're different chemicals that are used in cell-to-cell signaling, and we'll get into that in a later video. So now that we've talked about the endoplasmic reticulum, let's actually take a look at what it looks like. You can see right here that you have the nucleus, our old friend, right there, and all surrounding the nucleus you have a whole series of labyrinths, which right here is the endoplasmic reticulum. Now, blown up version, you can see it over here. You can see the rough ER, which has a bunch of ribosomes slapped on them effectively. So it kind of has a very rough, rigid form. Where the smooth ER, which you can see right here, has a really nice smooth texture to it because it's lacking those ribosomes. Now, your ribosomes right here, these are just kind of free floaters. They may end up being stuck to the rough ER. They also may not. They may just be hanging out in the cytoplasm looking for something to do. Now, like I said, this is a really good cartoon. Both of these things are. But it doesn't really show up that way in real life. In real life, you can see the rough ER right over here. You see these lines, which are actually membranes that are studded with those ribosomes. And you can see the smooth ER is over here, since you can see how that endoplasmic reticulum is very smooth and streamlined and doesn't have anything else going on with it. Now, we talked about the nucleus, we talked about the ribosomes, we talked about the endoplasmic reticulum. Now we're going to talk about the Golgi apparatus. Now, the Golgi does some interesting things. Pretty much vesicles, which are they're almost like soap bubbles. They contain newly made proteins. They go through the entire cytoplasm from the endoplasmic reticulum, and they end up at the Golgi apparatus. Now, the Golgi, it's a really interesting set of flat membrane-bound discs, and all Golgi does is packaging and distribution. That's all it does. It sends these proteins either out of the cell or to other locations in the cell. They may end up in the membrane. The Golgi will actually change these proteins around to figure out where exactly they're supposed to go and help them get there. You can see a picture of a Golgi right here. You can see the nucleus right over here. Right here we have our endoplasmic reticulum, and then these kind of multi-layered discs right here. See them there, you can see them there. Those are the Golgis. Now those Golgis, they can hang out in a number of different places in the cell. And what happens are the proteins enter this way. They go through the entire Golgi and they leave as vesicles, which are these things right here, these kind of balls of oil. So, to recap, we talked about the nucleus, which of course holds all the DNA, which are kind of like the blueprints of the cell. We talked about ribosomes, which are the workhorses of the cell, and the ribosomes, their whole job is to actually build and make proteins. We talked about the endoplasmic reticulum, which come in two varieties, the rough endoplasmic reticulum, or the rough ER, and the smooth ER, or the smooth endoplasmic reticulum. And the ER, its only job is to hold a bunch of ribosomes and make sure proteins are manufactured and changed and things like that. And then, of course, you have the Golgi, and its job, of course, is like being UPS. It packs different proteins and sends them either to other places in the cell or out of the cell. Tune in next time, and we will be talking about specialty organelles. This is Mr. Messina signing off. Have a fabulous day.